And what I'm trying to do is always with the same idea of application, that is towards therapeutics, but little by little I also got interested in evolutionary biology, because when you study cancer and the evolution of cancer, then little by little evolutionary biology, and then what is now called the philosophy of cancer is something that must be considered. So it's what I will present is something that has been designed about math applied to biology and medicine. Within this INRIA team, MAMBA, modeling and analysis in medical and biological applications at Laboratoire Gilles Lyons, with colleagues initiated to this kind of modeling by Benoît Pertam, and with youngsters also, Rebecca, Tommaso, Alex, and with Emmanuel Trellard, who is now the head of this lab, LGLL, and Camille Pouchol, who was a student, a PhD student, who is now associate professor in Paris Descartes University. So what drove me to this kind of weird thing that I have put in my title, Philosophy of Cancer, is the cancer puzzle. That is, we need to go beyond intracellular signaling pathways when we want to understand what are the determinants of cancer and find them not only in single cells, but also in cells that are within multicellular organisms, because cancer is not a disease of a cell, it's a disease of a complex construction, which is a multicellular organism, in fact, an animal. And to this end, I will mention the atavistic hypothesis of cancer that was advocated by many, but recently by physicists Davis and Lineweaver and oncologist Mark Vincent, to go to an evolutionary origin for cancer cell populations. So this was documented by between species phylostratigraphic analysis, and more recently also by genetic analysis by an Australian team, Good, Triggers, and others. But this addresses the question of coherence, cohesion within and between cells and tissues within a multicellular organism. Why and how is it disrupted in cancer? Or in other words, as Thomas Prade puts it, what does this deunification of this individual that is cancer consist of? So to understand cancer, we have to go to the great evolution that is going back to billions of years to understand where the big evolution, the Darwinian evolution, led us to. But also, if we want to understand what is cancer at the time scale of a petri dish or of human life, then of course we have to go at lower time scales, but having in mind also that the genome of us, of animals in general, bears all the evolution that has been developed during this big evolution for about 1 billion years. And as will be more or less explained in the next slides, plasticity in cancer cells, that is epigenetic driven, epigenesis driven, proportion to reversal to a de-differentiated status, can be explained by this hypothesis, this atavistic hypothesis of cancer. And moreover, it can be represented by reversible plasticity in mathematical models, and what is more interesting, such models are compatible with optimal control methods and then applicable to therapeutics. So this was an introduction to tell you how I came to be interested in cancer as a physician and mathematician, and how it makes me derive little by little towards more general questions, in particular what I call, or what some people call, philosophy of cancer. So the main thing about cancer is that cancer cell population um, is how should I say, endowed with plasticity. Then I will go to modeling cell plasticity by structured equations. What are sources of plasticity? How a multicellular organism, what is it and how can be deunified? And finally, possible consequences for therapeutics. So one of the major concerns in oncology is drug resistance or drug tolerance. Metastases, of course, are something very concerning for us, but I will not deal with them in this talk. So established drug resistance may be considered by ecologists, theoretical ecologists, or evolutionary biologists as some kind of evolutionary rescue. I will try to show you on a slide what this may mean. And I will also show you experimental evidence of cell plasticity and reversible tolerance to drugs in cancer. So are genetic mutations always concerned by drug resistance, or phenotypic switches also may they explain some kind of drug tolerance? So as I said, pitfalls of therapy are certainly drug resistance, also metastases, and I will deal with this, but side effects are also important. And if you want to deal with therapeutics, then it is necessary to take into account both side effects and drug resistance. And this is what we have done in a model with optimal control that was developed within the PhD thesis of Camille Pouchol. So genetically established drug resistance may also be favored by non-genetic, that is epigenetic adaptations, manifestations of cancer cell plasticity and driven by epigenetic enzymes. And so this can either be completely washed out, this may be completely transient, or it may persist and then lead to drug resistance in cancer cell populations, and maybe ultimately established as genetic mutations. Metastases are also something that we are considering, but not in this talk. So established drug resistance as an evolutionary rescue has been often presented in this nice figure by Ding in Nature 2012. And so enhanced heterogeneity in clones result in high phenotypic or genetic diversity of clones, and these clones may be eradicated by drugs, by therapies, or they may persist, and even new clones may appear, and this may be considered as a sort of evolutionary rescue. This is taken from a book by Stephen Jay Gould, citing itself biological evolution book by Price. And this is completely different setting. It's about evolution of species. But if you consider cancer cell populations, then they can evolve exactly with the same time scale scheme. That is, some environment is exerted on this population. Most of it is eradicated, except an extremely variable margin of the population that develops a new species or subspecies that is drug resistant, and etc. So this is what usually evolutionary biologists call evolutionary rescue, and it concerns cancer and its development. So mutations in this meaning are certainly underlying, and they remind us of Darwin's notebook, and they have been documented also in different papers, in particular this one by Carl O'Malley and Reeves in Nature. And so they're related to branching and mutations. But is it always so? Do phenomena linked to drug tolerance or resistance always rely on mutations? Well, not necessarily so. In this paper that was published 10 years ago, it can be shown that certain cancer cell populations, very aggressive cancer cell populations, here a non-small cell lung cancer cell cultivated, cultured in a petri dish, can be almost eradicated by a very aggressive chemotherapy, but not completely. 0.3% of the cells still survive. They just survive for some time, and after some time, with the same concentration of the drug, they thrive again. They develop colonies, and they can go back to the same kind of thriving colonies in the petri dish. But if you withdraw completely the bath, the very aggressive anti-cancer drug bath, then these very aggressive, very survival and proliferative cell subpopulation disappear, and you go back to the initial population. So this is a transient phenomenon, and moreover, it can be related to some epigenetic enzyme. And if you expose again the population to the same drug, the same scenario drives on again and again. So this is to show that drug tolerance is something that is not always, and most likely at first this is the case, it is driven also by epigenetic mechanisms that are transient. So to be short, drug persistence is when you go to drug resistance, established drug resistance, and drug tolerance is more the word that is used when this is transient. But as I said, tolerance can induce resistance also. And there are papers that show that genetic and epigenetic alterations are two sides of the same coin, as is advocated in this publication, for instance. Now, modeling cell population by structured equations is the job that we deal with at Mamba team, INRIA Mamba team, and Laboratoire Jacques Williams. So we are dealing with adaptive mechanisms that drive dynamics of cell populations, of cancer cell populations, towards drug persistence or tolerance. So phenotype structure cell population dynamic models are our everyday bread, if I may say so. And they make us able to study the asymptotic behavior of these cell populations by adaptive dynamic methods. And then it's possible by using optimal control strategies to apply this to therapeutics, as I will show in the end of this talk. So this structured cell population model, the first one that we used with two different traits, is published in a paper in Cancer Cell, in a cancer research, of 2015. And it's related to two traits that are related to symbolic traits or phenotypes, viability, that is expression of level of survival potential phenotype, and fecundity, the same, of proliferation potential phenotype. And introducing two different traits was the only way for us to represent the two-step observation in this petri dish that I shown to you previously. To be brief, without going deep inside the structure of the equations, It's a reaction, advection, diffusion equations. And it can be also represented by an agent-based model. And in fact, this was the work of two postdocs that we had, maybe some 5 years ago, Rebecca Chisholm and Thomas Lorenzi, and they showed nicely that all the elements of the Petri dish experiments were taken into account in the model with two different scenarios. The Lamarckian scenario, that is with adaptation of the cancer cells, and the Darwinian scenario with no adaptation at all. And well, we could study the heterogeneity and the adaptation of the phenotype. This is published in Cancer Research. And to make a long story short, we asked three questions to this model. Is non-genetic instability, that is in terms of the PD, the Laplacian term, the diffusion term, crucial for the emergence of DTPs? The answer is yes. What can we expect if the drug dose is low, which is something that is very often performed in experiment in labs where they deal with the drug resistance? Well, we can never obtain the two-step scenario in obtaining DTPs and DTEPs, drug-tolerant persisters, drug-tolerant expanded persisters. And genetic mutations can never explain this. So this was shown by simulations, not by a theorem in this paper. And so the conclusions are, should we choose between the Lamarckian and the only Darwinian scenario? Well, we can interpret our model results in the two cases, but non-genetic instability and selection are always mandatory. And if you have no DTPs present, that is no drug resistance, even at very low levels in the initial population, then you need to introduce adaptation. That is, you need to introduce the Lamarckian scenarios if you don't have any drug-tolerant persisters initially. No therapeutic consequences so far, maybe with epigenetic drugs, but they are still in their infancy. Now, to give you a more precise idea of what we are doing with adaptive dynamics and structuring populations with the traits. Sorry, I went too far. Let me put it in a very simple way. That is, we describe the heterogeneity of a cell population as a function of the question at stake. We always put a relevant phenotype, X here, which is for the sake of simplicity presented as a one-dimensional phenotype, but it can be of any dimension, in fact. It can describe by, let's say, coordinates in Rn, different settings that can be relevant to the description of population. And as you will see, it's possible to describe them with cell-functional traits, but some others do it when they're concerned with very limited questions with molecular traits. That is, for instance, the Q1 Geta1 in the celebrated Sui Huang and Tariq Enver modeling, with who are concerned with the choice between the red and the white blood cell lineage presented by stable dynamics at the molecular level in the individual cells. So this is not what we do. We just present abstract traits structuring the populations, but abstract traits that can be of any dimension. And here, the two main features of such modeling is that you can, by summing over the traits, follow the total cell population, but also at every instant in time, you can follow the distribution of the trait and its asymptotics. So the first possibility is following tumor growth, and the second is a more specific to adaptive dynamics and structured population. It's following the evolution of the adaptive distribution of the trait. And as I mentioned, space is not necessarily a relevant structure variable when we study drug control if we have no access to the geometry of the tumor. So what is the asymptotic behavior of the total populations? Well, it can be shown that, in fact, this is always with a very unrestricted hypothesis leading to convergence, that is to a plateau, a stationary value. What are the phenotypes in the population? What are the possible limits as a probability distributions for this probability measure? And all this, these two questions can be answered by using this kind of modeling with a structure of the population related to a trait relevant to the question at stake. And usually for our applications, we take the non-local Lotha Volterra model, where here you have the instantaneous rate, preferential rate, and here the instantaneous death rate, here the total population. So this is a non-local logistic term, and the whole here is the fitness of the total proliferation rate in the population, the instantaneous net proliferation rates. So, as I said, you can first obtain convergence, and convergence, it's quite easy to show that it's convergence towards the maximum over the x of the R of x over D of x. And it may be attained in different points that you are not guaranteed to have a direct measure, but it may be a sum of Dirac. But in simulations, it's usually a singleton, only one Dirac. And the non-local Lotha 1D model can also be used to study not convergence, but concentration in trait. And in this simulation, due to Camille Pouchal in his PhD thesis, you can follow the evolution of a trait here in a population with the same setting, in which you put initially a trait close to 1, and then you let evolve the population with time, and you obtain that it evolves towards a completely different trait, which is close to 0. So this kind of setting was completely essential in Camille Pouchal's thesis, and he also showed that with the help of Lyapunov functional, he could obtain convergence and concentration at the same time by using this Lyapunov functional and showing at the same time that this term goes to 0, this shows convergence, and that this time also goes to 0 with time, and this shows concentration on a 0 measure set of the traits space. So this is one of the first things that we can do with our modeling. And we most often use the non-local Lotha Volterra setting. Now, to go back towards biology, what are the sources of cell plasticity in cancer? Cell plasticity is not always pathological. It's certainly used in development, in embryological development, and even at adult age in tissue repair. So it's something that is completely natural, but among normal somatic populations, it's not so common. Tissue repair, okay, at adult age, development, certainly during embryogenesis, but normally not in somatic tissues, except the case of tissue repair. And this cell plasticity in somatic tissues is certainly loss due to loss of control on differentiations in cancer. Because in a normal cell, somatic cell populations, the differentiation is acquired and you cannot go back towards or de-differentiated stages of a cell lineage. But in cancer, this is different. I will show also that the idea of cancer stem cells is certainly interesting, but not necessarily needed, because any kind of stage in the differentiation trees can be a target for loss of control on this differentiation. So also cellular stress, and in particular the stress that is evidenced by chemotherapeutic drugs, can launch de-differentiation signal, and this has been recently documented in the general setting of cellular stress by recent papers. So development and tissue repair is certainly important, as I said. Is it due to how shall I say, cell-cell contacts, connections in particular, to make cell-cell communications possible, and this certainly uses energy, but this is not necessarily the only way to propagate the cell plasticity that is at work in embryology in particular. Plasticity is nothing at all, is not at all related to mutations. It is epigenetically determined, because you start from a genome when you have a zygote, which is perfectly defined, and you go with the zygote toward a completely differentiated multicellular organism, which goes only through differentiations which are only of epigenetic nature. But not in healthy cells, but in cancer cells, it is possible to go back towards de-differentiation in differentiation trees. Or you can also have access to trans-differentiation, which is even more complex. Here, the axolotl is an example in the animal kingdom of the possibility to regenerate a missing limb, and this is an illustration of animals, multicellular organisms, that can use cell plasticity for their own benefit without being suffering of any pathology. So I was mentioning that plasticity in cancer is actually loss of control of differentiation, and it's not only due to stem cells. For instance, in acute myeloid leukemia, it is well known that at rather evolved stage of differentiation, you have the blockade of this AML tree that is the acute pro-myelocytic leukemia, that is due to some molecular event, but it's not due to stem cell events. Also, uncontrolled differentiation can lead, and this has been studied by some people, in particular in London, about Barrett's esophagus, others about ductal carcinoma in situ, when there is an indeterminate histological zone, for instance, in the esophagus or in the duct, your cell populations are exposed to indeterminate histological fate and may lead to cancer. Transdifferentiations, as I said, so it's documented in a different paper. I will not go deep inside this.
Excuse me, can I
Yes,
ask for
yes.
some clarification for those of us that are totally ignorant? What exactly is the definition of plasticity? What has changed?
Plasticity, as what is changed is the control of differentiation. Differentiation in a multicellular organism is something very well controlled from the body plan until the totally differentiated cell types in the human species between 200 and 400 different cell types, but in very elementary multicellular organisms such as sponges, only 20 different cell types. But just the same, they constitute multicellular organisms, and they have been constituted, just like us, by cell differentiation on trees, starting from a body plan from the zygote and going to a very complex multicellular organism. And at each stage of differentiation, there is a control, in particular by the immune system, and maybe also by other mechanisms that are less well known. But I contend that this loss of control is precisely the nature of plasticity in cancer cell populations. So you can think of it as a quality of the cell, but in fact, it's a quality of the cell as at a given stage of differentiation. So it's a quality of a localized subpopulation in the development of a multicellular organism. So plasticity is a quality of a cell populations
So
that can be
it's like
considered.
changing the rules of operation. Is that right?
Yes, yes. It's certainly something. I mean, the control is in the rules of differentiation, but these rules may be lost, and this may lead, for instance, incompatibility between different neighboring tissues, which may lead, by loss of control on differentiation, also to loss of control on proliferation, and this may lead to uncontrolled tumors, well, the beginning of tumors, in fact. So, and this is also presented in a book by Marta Bertolazzo about philosophy of cancer. For her, the nature of cancer is both loss of control on differentiation and loss of control on proliferation, which I approve. But for me, I think that the loss of control on differentiation is, I would say, primordial. So also epithelial to mesenchymal transition is something that exists in physiology, but is also well known in cancer, of course. And it's related to the ability of cancer cell population to perform metastases. So plasticity in cancer, loss of control and differentiation in the development, but also in the maintenance of an evolved, of a finished multicellular organism that must be controlled at each step of differentiation. If somewhere it goes astray, then you may go to cancer. As I said, the cancer cells are not necessarily needed in the to obtain cancer, and this has been, as I said, in the promyelocytic stage of this form of AML, well documented. So stem cells are not necessarily present here. So differentiation control may be altered at any differentiation stage, maybe at stem cell stage, but maybe at many other places downstream. So what may launch de-differentiation signals, that is loss of control on differentiation? Well, cellular stress is certainly something that can do this, and to induce cell differentiation or de-differentiation, that is cell plasticity, which may induce what I may mention later, bed hedging in cancer, has been also proposed as a very important way to launch as a source of cell differentiation, which may lead cancer cell to differentiate in sort of bed hedging strategy. Differentiation or de-differentiation, because it is uncontrolled in cancer. So if you have any question, please don't hesitate at that stage. Yes, this has also been documented in this paper in PNAS of 2015 that showed that in multiple myeloma exposed to an anti-cancer drug, doxorubicin, it has been shown that cancer cells express cold genes that are ancient genes conserved in evolution, testimonies of a form of life adapted to high fitness under extreme stress. And these may be, for the cancer cell populations, the opportunity to develop bed hedging. But what has been shown is that these cell population, this multiple myeloma cell population, become resistant to doxorubicin as these cold genes are expressed. So what I contend is that the expression of this cold gene could help these cancer cells to launch de-differentiation stress-induced signal to the chromatin and then launch different phenotypes leading to different types of cells, some of them being resistant to the deadly insults that may be UV radiations, but toxic molecules, etc. Some others will be not, but the important thing is to let the genome survive. And then if just one subpopulation survives, then the genome of the cell population will survive. So as I also briefly mentioned, this speculation refers to the atavistic theory of cancer that I will maybe detail some more now. It's certainly best recently documented in the seminal paper by Davis and Lineweaver and Vincent also. Mark Vincent is an oncologist, Davis and Lineweaver are physicists, who advocates that cancer is a very primitive state of multicellularity that leads to non-cohesive states of multicellularity by lack of a coherent development program. But nevertheless, they try to reconstruct the multicellularity construction with, of course, being sure to be bound to failure. They use also genes that are in the genomes of us, for instance, or animals in general that have been evolving for billions of years, or at least 500 million of years, to their own benefit to resist a very aggressive environment. So how may a multicellular organism be deunified? This is one of the possibilities that atavistic theory of cancer be called for, and I will try to show you why it's interesting. I think that all of you have heard about the metaphoric Waddington epigenetic landscape, but I will also propose a complementary metaphor. And finally, I will, before going to therapeutics, try to present what of sort disruptions may elicit cancer. So the atavistic theory of cancer that was presented by Davis and Lineweaver and Mark Vincent in 2011 certainly relies on the fact that nothing in biology makes sense except in the light of evolution, that all of you know. And it's in this picture, it's related to the main development of multicellular metazoan capability that appeared about beginning 1 billion years ago, and our mammalian capabilities much more recently, maybe less than 500 million years ago. But the reversal that's advocated by this atavistic theory of cancer is that we go in cancer towards multicellular metazoan capabilities, but without the general organization. And this has been documented in a series of papers, in fact, ancient ones also, but they may have been forgotten, like papers of 1914 or 1996. But it's presently advocated in a very striking form, way by Davis and Weaver. So nothing in biology makes sense except in the light of evolution, but also nothing in biology in evolution makes sense in the light of systems biology, as Sui Huang put it, as I show you next. But also to mention something that made me interested in the billion-year evolution is the fact that the genes that have appeared in the development of multicellularity, as shown in these papers by phylostratigraphic analysis and in genetic analysis more recently, go back to this time of the planet development, of oxygenation of oceans and atmosphere, and the development of at the Cambrian stage of the evolution towards multicellularity. And what is convergent to this fact that multicellularity genes are also those that are altered in cancer, and to the fact also that there's a construction from elementary multicellularity, which consists of just cells without cell division, work division, that are just cell colonies, just communicating between them, and then division of work leading to, for instance, porifera, that is elementary sponges studied by Werner Müller, for instance. And after this division of work, epigenetic control of differentiation is the last stage of control. And this is precisely the inverse development of AML in fresh blood of patients suffering from AML that have been documented in this paper of Nature Communication. First, genes to be altered are the genes of epigenetic control, then the genes that are related to cell differentiations, and finally, the genes related to proliferation and analysis. So my here idea that reconstituted this phylogeny of the multicellularity toolkit that was developed at that time of oxygenation of the oceans and also of the Cambrian state should shed light on the robustness or fragility of genes altered in cancer. And as Mark Vincent put it nicely, attacking cancer on proliferation is precisely attacking where it is most robust. It's much better to attack its weaknesses, and in particular, adaptive immune cell immunotherapy is a much better idea. So, as I said, according to Sui Huang, who revisited the Waddington landscape, the epigenetic Waddington landscape that relies on genes. This is backstage of the epigenetic landscape, and this is revisited by a teach bifurcation epigenetic Waddington landscape by Sui Huang with bistable dynamics at the molecular level of each cell when they have choices to do between such valleys in the Waddington landscape. So nothing in evolution makes sense except in the light of systems biology for Sui Huang. So this is a certainly celebrated metaphor, and it has been used also or proposed, but it's just sketched by Tariq and Blair to in this paper 10 years ago to show that it might be not only presented on the celebrated Q1 GATA1 um, bistable dynamics that will lead to choice of either white blood cells or red blood cells. It can be put in all bifurcations of the, or it should be searched for in all bifurcations of the Waddington landscape. So I have also a complementary metaphor to propose here, which I call the wickerwork basket. So it's a sort of epigenetic landscape seen from upside down. That is, here you have the summit, which for me is a zygote with the body plan where stem cells lead to functional differentiation trees. These differentiation trees stem from the body plan, but they are controlled not only during the development of their lineage here vertically, but also at every horizontal stage. And this for me is what I call the cohesion watch. And this is a mission of an extended version of the immune system. And this view is coherent with views expressed by Thomas Pradeux in Philosophy of Immunology or the limits of the self, his recent books, for instance. And I say that it may be seen as a Borromean knot. That is, you need all of these. The body plan, from which functional systems or functional big functions of the organism stem, and these differentiation trees, and also the cohesion watch that should check at every stage that everything is coherent. That is the compatibility between the cell population that are developing in this representation of an organism, a multicellular organism under development. Now, what sort of disruptions may elicit cancer? First of all, fibers in this representation by my wickerwork basket metaphor can be fragile and break. This is what happens in the case of the promyelocytic acute leukemia, for instance. And so it's along the fibers that breaks may appear. And the cohesion watch, the immune system for me, should also take into account the control of the normal development along these fibers, these vertical fibers that represent the differentiation trees. But maybe more commonly, loss of control and differentiation may be due to impaired connections between fibers. And this is what I was alluding to when I was, for instance, alluding to ductal carcinoma in situ or Barrett's esophagus, when you have compatibility between cell population, neighboring cell population, that is not ensured by what I hypothesize as the cohesion watch that should be relying on the immune systems. So cohesion watch necessary to make the whole system hold together. And for me, it's a mission of the immune system. But not only the cell, the immune response to cancer or to pathogens in general, but something more that is essential to cohesion of the whole building. So as you said, as you saw, maybe modeling bed hedging in cancer cell can be represented with a two cell-functional phenotype for population heterogeneity. And this was the case with our modeling about transient drug tolerance phenomenon that we wanted to model from this paper of Cell, but it can be also thought of as driven in a heterogeneous population by not only a two cell functional phenotype, but a three cell functional phenotype that could be, for instance, adhesivity motility or fecundity motility, germinal somatic, which is at the basis of the development of multicellularity. It can be also thought of as a relying on an opposition between fecundity and viability, but also with a cell-functional intrinsic variable that is plasticity. So I propose, and this is a work that is underway with a PhD student, that bed hedging in cancer can be due to a divergence of phenotype where the plasticity of cells leads an important role. And also I propose that since cancer cell populations continuously reinvent multicellularity, that this may be also relevant setting to represent the origin of multicellularity. And this is an example of what Frank Alvarez-Borges has been doing with this simple reaction diffusion equation. With here you see the divergence of phenotype in this cartoon with time. Little by little, a single phenotype divides into two. And finally, it goes to extreme values of the two phenotypes, X and Y, that can be thought of as fecundity and viability. And this is accompanied by the fact that there is a very precise concentration of the plasticity phenotype that was first around 0.5, and here that is concentrated around 0.25. So this is underway, of course. But for therapeutics, what can we say? So, Benjamin, how much time have I still got?
Yeah, you're actually a little bit out of time, but okay.
So
Yeah.
I will just sketch briefly what can we,
All right.
what can be designed for therapies.
All right, great.
So, well, I was just saying that insufficiency of a single cell-based therapy has been documented in many papers. But about cell-killing strategies, as I maybe have said earlier, it's important to take into account both what you do to the cancer cell population and what you do to healthy cell population. And this is what we presented here in this paper that was published by with first author Camille Pouchol. We have two populations, healthy and cancer, with exactly the same structure. And drug targets here is an added term represented cytotoxic drug, and here just a drug target representing a cytostatic drug which slows down the cell cycle growth. And we want to minimize the final final horizon time state of the cancer cell population. So this is a theorem that I will not dwell into very much. One of the nice features of this model is that it shows that if you exert very important, very high and constant doses of drugs on the same population, you're certainly doing the pessimal strategy. That is the worst thing that you can do with the development of a resistant cell population and the healthy cell population that will go to zero. So there's a theoretical theorem that is illustrated here. And in fact, just consider that you have this optimal control strategies that is represented here. These are the optimal U1 and U2 drug concentration. And this has been published in this paper. Yes. Just know that the strategy, drug holiday in oncology, in the clinical oncology, is called such so because you do not do anything with the cytotoxic drug to let the cell population become sensitive to the drug. About immunotherapies, we have just done this. This is underwork also to represent immune checkpoint inhibitor therapy. So this is also something that must be developed. And about my metaphor of the wicker work basket, if there is such a thing as a cohesion watch, then we should, assuming the existence of this cohesion watch, we should look for intercellular connections during development that are altered in cancer, and how can these intercellular connections be re-established. This could lead us to non-cell killing anti-cancer therapy, a goal that is not unreachable. So this way of conclusion is that we should look for new therapeutic tracks in cancer. Immunotherapies are certainly a promising track, but we have not understood all of them, and many things remain to be done about that. And what I propose in maybe a distant horizon is try to understand what a multicellular organism is constructed with, what is its coherence, and how such cohesion is altered in cancer, and how can it be re-established without killing cells. So these are some references from cell plasticity to philosophy of cancer, as you can see, which is one of my tracks of development, but in modeling, but not only. So it's also modeling by PDs, as you can suppose. So I thank you for the attention of the audience, and ready to take questions, or by mail, of course, if you prefer.
Thank you, Jean, for this very complete talk with both math and applications and philosophy, actually. Yeah, so if there is questions, please go ahead.
So in this cohesion watch, suppose you detected some loss of control in the in differentiation. Is there any way the body can fix it, or does it have to be in medical intervention? I mean, and on what levels does the fix take place?
Well, what I have in mind is firstly trying to understand what are these compatibilities between cell subpopulations that are mandatory for us to be constituted as a coherent organism. I would say coherent for signals and cohesive for tissues. And if we can understand what comes out in development, but maybe as some of you may have followed works by Thomas Lecuy. There are two ways to understand this, and they are complementary. There is a biochemical program that is contained in the zygote, which makes all of the development of the embryogenesis towards a finally developed animal possible. And this is also possible as a set of equations, in fact, because you have matter on which to develop the self-organization, which is what is usually studied by embryologists and physicists, biophysicists of development. So it's both at the biochemical, that is at the stage of the program, as much as it can be understood, and also at the embryogenic state of development, that is a more, how should I say, a biophysicist deal, in fact, that both should be studied. Some people have begun to study, but this is very difficult, I know, these genes that are related to compatibility of subpopulations in multicellular organisms. And I think that by studying these intercellular communications that appear during development, it could be possible to act on them, that is on what I call the cohesion watch, without being compelled to destroy cancer cells. Because cancer cells are, if you reestablish a cohesion that is locally lost in a cancer that's beginning its development, the cancer cells will not follow their development, as is the case, for instance, in the treatment of acute promyelocytic acute leukemia, in which you give both therapeutic that reestablished the differentiation by leveraging the blockade of the differentiation tree, and simultaneously you give an anti-cancer drug. So the surviving cancer cell population will be eliminated, and this can be be done by using anti-cancer drug. But most importantly, the cohesion watch should be reestablished by other types of drugs or other therapeutic means that are, except in very specific cases, not known. And